today we are doing this thing, that is storage and storage of data, as well as how to retrieve DNA sequence from the database. What is database, or what do you mean about the database term? So in case of bioinformatics, we have to make certain place where we can store or we can retrieve certain kind of data. Can anybody tell me the difference between these four terms? One is known as data, second is known as information, and third one is known as knowledge, and fourth one is known as intellectual. Any fact, figure, picture, sketch, map, anything can be data. Either it can be informative one, or it cannot be informative, non-informative one. So we are saying that the collection of anything, if we are saying in the layman language, that collection of anything is known as data, or in terms of scientific terms, we can say that the collection of informative as well as non-informative thing, relevant or irrelevant thing is known as data. So in case of bioinformatics, we can collect any data. For example, either DNA, either protein, either metabolic pathway, anything we can collect. It can be correct, it can be wrong also. So it is nothing but the data kind of thing. If we are filtering or we are removing irrelevant data, I am only getting the relevant data or the informative thing, then it becomes information. When we filter data, then it becomes information. It only consists of relevant data. In this case, for example, I am talking about the information thing. After that, if we are again filtering this information, then we get knowledge. And if we are using that particular knowledge, then it becomes intellectual thing. So it is nothing but the knowledge database, which we are considering in bioinformatics. The bottom one is known as data, which is in large amount, or the most of the amount is present in this, that is known as data. If we are filtering this thing, then it becomes information. If we are again filtering this thing, then it becomes knowledge. And if we are again filtering this thing, then it becomes intellectual thing. So in case of bioinformatics, most of the work is doing here. We have to store something. After that, we have to find out the relevant thing. And using that particular information, we have to increase the knowledge of the database. For example, if for example, someone has said that, sir, this particular G1 gene is present in Homo sapien. Okay, I'm saying that this is nothing but the information for me. Whole DNA is the data for me, and this, you are picking certain relevant information, then it becomes the gene, and it is nothing but the information for me. And someone said that, sir, actually this gene is responsible for glycolysis. This is nothing but the knowledge for me. So you are going from the bottom to the up. And when you go from bottom to the up, then the amount of that particular thing going to decrease, and it resulted into intellectual thing. And you can say that in case of glycolysis, this glucose can be converted into pyruvate. All the things are there. All it comes under this intellectual kind of thing. So in case of bioinformatics, we are dealing with these four levels. That is mainly at the information and knowledge level. That how can we transform data into knowledge or information. So in this case of first experiment, we are using three different databases. Have you heard about this database? NCBI, EMBL, and DDBJ. During 1990s or during 1988 or 1990s, US government has said that they have to sequence the whole genome of that particular Homo sapien. So they have started a project regarding that, and the name of project is Human Genome Project. After sequencing in the initial years, they said they observed that the data is very large and it is coming in the huge amount. So they have made a database, and that database is named after that, is known as NCBI, or National Center for Biotechnology Information. I'm telling you a story, a brief story or hypothetical story, so that you can easily remember these names, because they are very important in bioinformatics. So first, NCBI have been made, and for example, during 1990s, they have made, and they said all the gene related information or the DNA or RNA related information should be come under this. That is NCBI, National Center for Biotechnology Information. Whenever you made certain kind of servers or database, you need funding. For doing that thing, NCBI was seeking out certain kind of funding. They got two different agencies, US agencies, and these names are NIH and NLM. Okay. For example, there is a, have you heard about this term, NIH? They said that you are making this thing, we can fund this thing, but you have to maintain our data also. You are making, you are doing on Human Genome Project or any other project, you are sequencing and you are taking your data in that particular database, NCBI. But if I am funding this thing, you have to keep their data also in that particular database, NCBI. So this is NIH, National Institute of Health. Along with that, there are certain doctors or medical professionals who said that if you are making certain database, then if we paid you, then you can also take our data in a confidential manner And this NLM is okay. This is National Library of Medicine. So all the professionals of medicine keep their data in this. That is NLM. PubMed is the repository where all the medical journals are available in this case. So these three agencies collaborated. One is NCBI, another is NIH, and third one is NLM. So they make a website or they make a database. Right now it is there. It is free of cost to everyone. So they make that ncbi.nlm .nih.gov. Gov is for government use, or you can say open to all. So we have made this thing, and this is the website of the NCBI. NCBI, NLM, NIH, and GOV. So here the first repository which have made by NCBI only, National Center for Biotechnology Information. The funding is given by two different agencies. NLM, which maintain the publication data, that is National Library of Medicine, and this is the health related data, that is NIH, National Institute of Health. All the information are coming in this particular thing, and they have said that all the gene and gene related information should be come under this, that is NCBI. So first database I have talked to you about, that is NCBI. It is related to genes or DNA or RNA, anything related to genome, genes, nucleotide, anything is related to that. Have you heard about this thing, that is a search engine? For example, in case of NCBI, if I'm considering as a browser for gene, DNA, and RNA, all the things are available, and the search engine for that is Ontrace. Name is Ontrace. So whatever you want to search related to DNA, RNA, protein, not proteins, related to that particular gene sequence, then Ontrace is very useful search engine for browsing in that particular NCBI. For example, as I earlier told you, US has done all the things related to genes, DNA, and RNA or nucleotide sequence. After that, in case of central dogma, what has been left? This is DNA, this is RNA, and this is proteins. So in case of this, there is an agency known as EMBL. This is a hypothetical story. I'm just saying that so that you can cover whole molecules in case of central dogma. So in case of EMBL, what is the full form of that? So EMBL is European Molecular Biology Lab. In this case, all the protein or amino acid related information is there. It is built by European Union. All the things are available. So for example, in this case, the browser is NCBI and the search engine is Ontrace. In this case, what is the search engine? SRS, Sequence Retrieval System. So in case of EMBL, all the searches are performed by this, that is search engine, that is SRS, Sequence Retrieval System. After that, Asian countries came, and Japan is the major prominent country at that time during 1990s, and they had made a database that is known as DDBJ, DNA Data Bank of Japan. All the kind of metabolic information or internomics information are present in this particular database. The good thing about research is that when you collaborate, you will get good results. So in case of this, because DNA is connected with RNA, RNA is connected with proteins, and proteins are doing certain kind of internomic study or metabolic things they are doing. So we have to connect these things. So these databases are connected to each other, and they share their information periodically, or you can say bi-yearly or quarterly. All the things they share, all the information are shared by this particular database. So either you can go from this, you can search a gene from NCBI also, you can search a gene from EMBL also, you can search for DDBJ also. But specifically, if you want DNA sequence, go to NCBI only, not go to DDBJ. For example, there is a database known as KEG, Kyoto Encyclopedia of Genes and Genomes. It is maintained by DDBJ only, and it is related to metabolic pathways. So if you want to study certain metabolic pathways, go to this database, that is DDBJ. It does not mean it does not consist of any nucleotide sequence. It consists because it exchanges the material, all the data from that NCBI. It also consists of protein information because it exchanges the data from EMBL. But it has certain kind of ID that is linked towards either EMBL or either NCBI. We are first discussing what is the classification mechanism behind this database. So according to computer science, we can classify the database into three different types. First is known as primary database, second is known as secondary database, and third one is known as composite database. So in case of this, this is known as raw data. For example, it may be right or it may be wrong, or relevant or irrelevant data it may be there. So it is nothing but the primary database. For example, Siddharth has done DNA sequence. He has written AA, TT, GGGG, all the things is there in a notebook he has done, and he has submitted to NCBI. All the things is there. So it is nothing. It may be correct. It may be not correct. So it is nothing but the primary database. When we filter this thing, or I have told you regarding this database or you can say knowledge tree. In this data, when filtered, it becomes information. So when you filter this thing, then it becomes the secondary database. Or if you are combining both database, that is primary as well as secondary, then it becomes composite database. But there is another definition for this, that is database. According to database, in case of biology, we are classifying it into two categories. First is known as sequence database, and another is known as structural database. In case of sequence database, it can be DNA sequence, it can be RNA sequence, or it can be amino acid sequence. While in case of structure, it of protein structures. Either it is a primary structure of amino acid, or it is secondary structure. Secondary, it means the purest form, it is the raw form. Similarly, in case of sequence, there are two different kind of sequence. First is known as primary sequence database, and second is known as secondary sequence database. All the experimental work, all the raw data that comes immediately to the database is known as primary sequence database, and it comes under either NCBI, EMBL, or DDBJ. Any work which you have performed in your laboratory and you want to submit it, then you have to first go towards either of the three database, either NCBI, EMBL, or DDBJ. That's why it is known as fundamental database. Fundamental database, or it is also known as primary sequence database. So this kind of thing is there. Primary sequence database is also known as fundamental database, and the name of these databases are NCBI, EMBL, and DDBJ. We are working on this particular database that is known as NCBI. While in case of secondary, if we are filtering something and it becomes this secondary sequence database, it may consist of amino acid, or it may consist of filtered DNA or RNA sequence. While in case of structure, if you have performed certain kind of X-ray crystallography experiment or NMR spectroscopy, and result has came, and you are storing somewhere, and that place, that database is known as primary structure database. While if you are filtering or choosing only relevant data, then it becomes secondary structure database out of that. I have told you regarding the simple definition of database. One is primary, secondary, and composite database. Now the refined definition is primary sequence database and structured database. Now I am going towards a more complicated one, which you have to study regarding that. This thing I have showed you in the paint thing. That is, in this case, for example, database is there, either sequence or structured database. In case of sequence, both primary and secondary database are present. In case of structure also, primary and secondary structured database are present. You have to at least name, remember this name, because sometimes the question has been asked, enlist any three primary structured database that are present in that. For example, in this case, NCBI, National Center for Biotechnology Information, EMBL, European Molecular Biology Lab, and DDBJ, these are known as fundamental database. While in case of this, if I am filtering this thing, it becomes secondary sequence database. This is TR. TR means translated EMBL. When you translate DNA or when you translate mRNA, then it becomes protein. So it consists of protein thing. So in case of secondary sequence database, it consists of protein database. The name of that is translated EMBL, that is European Molecular Biology Lab, or it is also known as UniProt. Unique protein, KB means knowledge base. So all the things are available in this case. While if you again filter this thing, if you are making structure from the sequence only, it may be correct, it may be incorrect, or you are using X-ray crystallography or NMR spectroscopy, then these kind of database are available for you that will provide either relevant or irrelevant information. That is CAT plus class, architecture, topology, homology, scope, structural classification of proteins. And if you are again filtering this thing, you are going from raw to the pure form. Here it is a purest database, that is known as PDB. That is known as protein data bank. There are three different coordinates of proteins that are available, and it is the purest form. It is present at RCSB, that is Research Collaborative Structural Bioinformatics. It is a database where all PDB files are available. I will show you. And this is present at NCBI, that is Molecular Modeling Database. All the three-dimensional structure of proteins are available in this. So someone can say that which is the raw form. This is the raw form. It is also known as fundamental database, and it is the purest form, that is secondary structured database. All the database are present in bioinformatics only. Okay, so I'm going to the major definition of this database. These are there. It is a complicated one, but you have to at least remember which kind of database it is present. For example, I have told you regarding database. In case of bioinformatics, specifically, first is sequence database, second is structured database, and third one is functional database. In case of sequence database, there are two, primary as well as secondary. Someone has told me about the raw thing and the purest thing, or relevant and irrelevant thing, that is raw thing, or if we are filtering that thing, information kind of thing, then it becomes secondary. Similarly, in case of structure, primary and secondary is there. But in case of functional, various kind of specific organism database are there. For example, model organism database, those organisms that share certain kind of genetic information with, you can say, homologs, that are homologs to that particular Homo sapiens, then they can consider as model organism. There are various examples like rat, mouse, or chimpanzee are there, or macaque are there. Certain examples are there which can be considered as model organisms. Specifically, mouse and rat are there. So in case of this functional gene expression database is there, phenotypic database are there, protein interaction database, signal transduction, metabolic pathway, specialized, for example, they are performing certain kind of interaction kind of docking kind of studies are there. So these are specialized kind of database. And last one is additional database. So in case of this primary database, primary sequence database, I have told you regarding that, it can be of nucleic acid, it can be of protein. If it is nucleic acid, it can be either of DNA and RNA. But if we are specifically bind towards this fundamental kind of thing, which I have told you regarding that, primary sequence database is known as fundamental database. If we are going toward this category of DNA, then these are three databases that are commonly found, that is EMBL and DDBJ. These are the three databases that come under this primary sequence database, specifically in DNA. So anything related to nucleotide, I think in the central dogma thing, you have heard about this AUG. So it is present in mRNA sequence because U is present. So I think it is present in mRNA sequence. But in bioinformatics, we are not considering that thing. Rather than using mRNA, we transcribe it back into cDNA, and we are taking only DNA sequence. If it is DNA sequence, then it becomes ATG. It is nothing but the start codon. So in case of this database kind of thing, we are generally considering DNA as compared to RNA. But there are various databases that are available for RNA also. For example, mirbase is there. Other than that, there are other databases are also available. For example, RFAM is there. All the databases are available. You do not have to remember all the names, but you should know what are the various types of databases that are present. You should know at least the name of certain database. For example, in this case, I have told you about NCBI, EMBL, DDBJ. Here I have told you about mirbase as well as RFAM. Here, for example, protein primary sequence database in case of protein. These are TREMBL and UniProt is there. For example, there are certain kind of secondary sequence database which are filtered form of this particular primary database. These are 23andMe database is there. Other than that, there is HapMap, haploid mapping. Other than that, there is OMIM, online Mendelian inheritance in men. Those diseases that follows Mendelian pattern in Homo sapiens, that are nothing but OMIM. That is online because it is available online. First, earlier it was MIM, Mendelian inheritance in men, but right now it is available online, so it is nothing but OMIM. It is present at NCBI only. So there are various kind of database that are available. You have to primarily focus on these two, which I have told you in the last slide. That is this. These at least database you should know about what are the major types of database that are available in case of bioinformatics. So primarily we are today focusing on this thing, that is NCBI. I have told you I have shown you also that what is NCBI.